that's working. Yeah, that seems to be working. All right, let's see. Have we gotten some people? Look, we've probably lost a few today, but that can't be helped. They'll have to watch the recording later. All right, I'll get started. As per usual, if you got questions, just interrupt as we go. And yeah, let's do it. So this paper is one, I think it was originally recommended by John Smart through some searching of his own. Came out pretty recently, came out November last year. I feel like we got a lot of papers from late last year, 2024. And we're back in the motor cortex instead of the hippocampus today. So let's see what we can find from the cortex. If my slides advance. There we go. So just as a reminder, although I think all of you know this pretty well by now, we're here reviewing papers because we are interested in the memory decoding challenge, which is where what we want to know and what we want to see is the first demonstration of a decoding of a non-trivial memory based from the static map of synaptic connectivity, or the connectome. Can we pull memories out of static brain structure? So that's the big prize, and then the little prizes are each year we aim to have these 4 annual research awards where we honor and highlight research into how learning and memory are physically encoded in the brain as steps towards this ultimate goal of being able to read memories out of static brain structure. And the paper we're covering today, as part of that, we're going to have this ceremony at the Society for Neuroscience event in San Diego. It's going to be on Monday, the 17th of November in the evening, which is in the middle of the conference. We're going to give the exact venue when we get that very soon. But if you're going to be at SFN, you should definitely come along to this. And if you know anyone who's going to be at SFN, you should definitely get them to come along because it'll be fun. There'll be a panel, 2 panels, one for carbon copies sort of stuff, one for aspirational neuroscience. There'll be some free drinks. It'll be a good time. Talk about memories. Okay, so some background. This paper is examining motor behaviors and specifically looking at like how different motor behaviors are encoded in the motor cortex. And in particular, they're focusing not necessarily just on the actual activity that drives the motor action itself. So like activity that happens during muscle extension, but they're also interested in preparatory activity. So what's happening in the motor cortex just before an animal reaches for something? How does it work out what it's about to do? Now, the question that they have is, there's a lot of actions where you do the same thing, like you reach out to grab something, but with different contexts and contingencies. Maybe you're grabbing something in a game, or you're grabbing a door handle, or you're grabbing a weight or something. And you can have these different contexts and different values and contingencies that drive an action with exactly the same kinematics to it. So the question is, to what degree these different activities are represented differently in the brain? And also, is the activity that drives these different representations of these different motor actions in these different contexts, are they static across all sorts of different contexts and all sorts of different time points? Or do the neurons that are driving that activity change over time? Do they undergo some sort of representational drift? And this is an interesting question because there have been a number of studies of looking at representations of particular sensory states in the cortex, which have shown that the neurons that are encoding a particular sensory state, let's say, I don't know, seeing some object, if you look at them over time, they seem to shift to a certain degree. So it's not necessarily the same population of neurons that's always firing when the same sensory stimulus is happening, or sensory representation, or something to that effect. I will admit I'm not fully on top of the representational drift literature, which will come up more as this talk goes on. So if you know more about it, please correct me. But the idea here is, like, over a long period of time, is it the same neurons encoding the same information, or is it somehow changing in some way over weeks and months? And then the last thing as well is, to what degree, when an animal or human learns new motor skills, do the old representations get changed by the addition of that new information? So is the population of neurons that are encoding a particular motor memory, are they influenced, altered, manipulated in some way as that animal learns more and more motor engrams that it's shoving into its motor cortex? So that's some of the background. Let's get into this specific paper. So it's a combinatorial neural code for long-term motor memory by a group in, sorry, I've forgotten exactly, but I do have it up here, somewhere in the US. A group in, if it will load, my computer's being slow, in Durham in North Carolina and Baylor in Texas. So let's do it. Okay. Apologies. Come on, next slide. Next slide. Okay, cool. So the particular task that they are using in this experiment is a directional licking task. So basically they train these mice, they're water restricted, so that they want to somehow get their water. And the way they get their water is they have to either, they have to put their head into this little hole and they lick left or they lick right. And if they lick correctly, they get a water reward under particular task conditions. And you can see here, they've actually done it so that they've set it up in their home cages so they can enter and leave at any particular time to do the task and get a little bit of water. And they do this task repeatedly over a long time. So this is over 2 months, and you can see that they're self-initiating when they do these particular little bouts of their little licking experiment. They're doing this in the dark using infrared, and they're tracking the position of the mouth of the mouse. And the specific task that they're eventually training them up to do is there's a little pole that's marked here in either purple or in green that's either further back or it's further forwards. And what happens is at the start of any particular trial, this pole gets extended and it brushes their whiskers either at the back or at the front. And in this particular task contingency, if it brushes their whiskers at the back, that means they have to lick to the right to get water. If they lick to the left, they get no water. And if the pole is instead at the front, instead of at the back, they have to lick left to get water. And if they lick to the right, they get no water. And the way any individual trial works is there's sort of a sample period where the pole is extended for 1 and a bit seconds. Then there's a delay during which if they lick, they don't get any water. So there's this enforced delay. And then after 1.3 seconds or so, there's a go cue, a little sound, which plays, and then they have to respond. And they lick left or lick right to get their water based on the position that the pole was in. What's going to make this more interesting is after a certain level of performance, they're going to start to reverse the contingencies for the task. So you can see once they start performing above 75% performance, then they will switch it around so that instead of licking right if the pole is at the back, they're now going to have to lick left if the pole is at the back, and vice versa. And they do this repeatedly with the mice once they start reaching plateau performance, or once they start reaching about over 75% on these trials. One thing I will note, as someone who's run these sorts of experiments on mice, is you will not believe how dumb mice are and how many trials it takes them to learn these particular tasks, where this takes days and days and days and like thousands of trials for something that a human would learn in like 3 or 4 trials or something. It's incredible. But it's both incredible how long it takes them and that they do actually learn it at all. That's just for me having done this. Anyway, one of the things that they also note is despite the fact that they're going back and forwards and they're learning this reversal contingency, the actual motor actions that they're taking when they lick left and they lick right become extremely stereotyped after learning the task initially. These are just some like sort of trajectories in red or in blue for licking left versus licking right. And you can see their velocities and their like directions become very, very stereotyped. So that they only note that because what they're going to show is that the responses become pretty much the same constantly, but the activity that's happening in a preparatory manner is what's going to change and be interesting. So one thing they also did before we get into the more interesting experiments is they were wanting to check whether the activity in the anterior lateral motor cortex was actually important in enabling task performance. So they took a GAD2 Cree mouse, which is, I think, a GABAergic mouse, as in like they can use channel rhodopsin. There's channel rhodopsin in GABAergic neurons, so they can drive activity of the inhibitory neurons using a red light. And what they're going to do is they're going to essentially, during either the sample period of time, so like just when the animal gets exposed to the pole, which tells it whether it's supposed to lick left or lick right, during the delay, or during the response when it actually has to lick left or lick right. On trials where it is instructed to lick right, which is when they're going to shine the red light on the left motor cortex, and they're going to see if that impairs its ability to perform the task. Because if the left motor cortex is inhibited, then it should have difficulty licking towards the right. And you can see here that indeed their percentage correct rate goes down dramatically on trials when they've had this optogenetic stimulation in a dose-dependent manner, depending on how much light they have. And they see this sort of the biggest effect during this delay period of the experiment. And so from this, they're going to be focusing on the delay period primarily during the rest of the experiments that I'm going to be showing. So they're basically saying that the activity in the ALM is important for task performance, particularly the activity that's occurring during the delay. So after the mouse has been exposed to the cue that tells it if it's supposed to lick left or lick right, but before it's actually made that response. At the bottom, you can see an example of like how that actually happens during individual trials instead of just averaged, where you can see they've trained this mouse initially to do a set of different reversals, and then they've sort of left it past peak performance for a while. So they've let it plateau, and then they're just showing the optogenetic stimulation during this plateau, and you can see it basically knocks the animal back down to chance levels when it's trying to do the task. Can everyone see my mouse, by the way, when I move it around on screen? Or is that invisible? Yes, you can. Okay, great. Okay, so then we're getting into the more interesting questions. The first one is, over time, are the same set of neurons responsible for encoding this sort of preparatory activity on whether the animal's supposed to be licking left or licking right and remembering the task contingency? Or do these neurons change over time? So how they're going to do that is they have these cranial windows above the ALM where they're tracking a whole bunch of different regions. So several different regions for each different mouse. From memory, there's about 8 mice, and there's, I think, something like between 6 and 9 regions that they're tracking per mouse. And they're looking at the positions of individual neurons in the motor cortex that they can see. You can see they have these initial images, what it actually looks like on the top. They're sort of tracing out the individual neurons that they can detect, and then they're matching these neurons across different sessions. And these are some example sessions of a session on day 1, so the first day of tracking, day 17, and day 60. But the point here is that they're pretty good at tracking these individual neurons over even months, and being able to look at the activity of these neurons over months. I didn't make clear these are genetically calcium fluorescing animals. They're like Thigh1 GCamp something, I forget the exact name. So they're able to see the calcium fluorescence in these cortical neurons during the trials. And so this is an example of the actual activity that they see from these regions over time. Here you can see this is on day 1, this is on day 13, and this is on day 33 for animals that are licking left versus licking right in particular regions of interest. On the top, you can see particular traces for activity for a distinct neuron in those different regions on those different days, just to give you a sense of the change in fluorescence over the course of the sampling, the delay, and the response. And you can see, for example, the neuron on the bottom really fires a lot, has fluorescence during the delay period. But the more useful thing to look at are these heat maps. And you can see this is one region where you're seeing neuron 1 through 386 sorted by when they fire during the sample, the delay, and the response period. And again, it's the left is the neurons firing on day 1, the neurons firing on day 13 in the middle, and the neurons firing on day 33 on the right. These are all with the same sorting. So it's the same neuron at the top for day 1, day 13, and day 33, which is important because what you can see is that on day 1, day 13, and day 33, these neurons are all pretty much the same neurons. So they're showing that the firing patterns are the same across time, that they haven't seemed to switch. So if you're a neuron that's firing during the sample period on day 1, you're probably going to be firing during the sample period on day 33. And what they also show here at the bottom is if you look at the neurons that are firing in a selective pattern, so they're firing in particular when the animal is supposed to lick left versus to lick right, you can see the distributions of how selective those neurons are of the ones that are selective. So they're not looking at or showing the activity of neurons that are just unresponsive to the task in general. You can see that there's neurons that are selective during the each of these three phases during the sample, the delay, and the response periods. And you can also see that when they go from being experts in the early phase of that, so at the start, and also later, you can see the same essential pattern of neural activity. So all this is just to show initially that the neural representations during the delay period and other periods of which direction to look at seem to be quite stable. They're not drifting, at least in this particular experimental condition. Make sense before I go on? I'm not seeing any hands. Cool. All right, great. Okay. Oh, Ken? Yeah, Ken, go for it. You're muted, Ken. I can't hear
you. Sorry. These particular mice here that you're showing on this slide, they had only learned in quote-unquote one context?
So I think these mice may, I can't remember whether they're like this one on the bottom, where they've only learned in, this mouse on the bottom has learned multiple contexts, but then it's being trained up on one and left on one for a while. But I think these mice here have only learned one context and haven't been reversed yet when they're being examined in this particular experiment. But I would have to check the paper because they do a bunch of these different variants. So my apologies for not remembering the specific one. All right. So then the question is, what happens when you start changing the context and you start changing the contingencies so that they, or at least I think that's what's happening in this particular experiment. Let me remember. Maybe that's not what we're doing just yet. So I think in this particular experiment, what they're looking at is to what degree can they classify, can they essentially develop a decoder that classifies whether the animal has licked left or licked right based on the activity that they're seeing in the cortex. So basically what they want to know is they're looking at the firing of these neurons in the motor cortex, and they're looking for a signature that determines, hey, the animal has licked left or has licked right. And they're basically primarily interested in what is the difference between neural activity when the animal is licking left and licking right. So they look, for example, at the average neural firing when the animal's looking right, so this right vector, and they subtract the neural activity when the animal's looking left, and they call that a coding direction during the delay vector. So this CD delay is going to come up again and again and again, and it's basically like the signature of neural activity that distinguishes licking left from licking right. And they want to see whether, like, looking for signs of this coding direction vector, this activity is reliably able to indicate whether the animal is going to have licked left or have licked right, to the degree that you can basically develop a decoder which will tell you, yes, the animal licked left or licked right based on that. And what they show here at the bottom is, again, yep, Ken?
Sorry. The yellow bar there is saying that they're only looking at the last half of the delay period for this?
Yes, exactly. So they're just looking at the activity during the late delay period.
Okay, and so I just want to make sure that, so the pole that they're sensing is already gone.
Yeah.
And they haven't licked yet. And so essentially the mouse is it's essentially the mouse is kind of thinking about what it should do, but it can't do it yet.
Exactly.
Okay.
Yeah. So the mouse is like, it's had its probe. It's like, okay, am I supposed to lick left or am I supposed to lick right? But it's not sensing anything at that point in time, and it hasn't started moving yet. It's, as you described. And there's activity that's happening in the ALM, so the anterior lateral motor cortex, and that activity is indicating whether, the mouse is like, I'm going to lick left to get my reward, or I'm going to lick right to get my reward. And what they're showing here is that activity in the ALM is very stable across time. So you can see at the bottom here, this is again measured in days. And this is trying to decide whether they can decode whether the animal's going to, lick left or lick right, and whether that is the accurate thing to do. And they're showing that they have these very high decoding accuracies, so around 80% or so, across all of these different sessions, and also within session. Yes, John?
Yeah, thank you very much for this. Are they picking that particular delay because it gave them maximum prediction? How do they come across the 1.3 seconds as the average for the delay?
I don't know precisely, but I can guess that they've picked whatever the minimum time is where they're sure. What would it be? So the animal,
it hasn't been able to give a response in less than that, basically, is what you're saying, right?
No, I mean, you could have it so that it responds immediately during, immediately after cue presentation. I think the key thing they want to do is they want to be able to distinguish, the activity that drives the actual motor response from the activity that drives the sensation of detecting the cue.
I get it now. I get it.
Yeah. So it's like
1.3 seconds to measure what's happening in those neurons, and that's why they're setting that delay at that particular time. They're habituating the mouse to have to wait that amount of time. I got it.
Yeah, and they want to distinguish it from the actual motor activity and from the sensation of the cue. And they need enough time to do the scanning of the motor cortex. But then they want to minimize it so they can do as many trials as possible without having to wait too long, would be my guess as to where that number comes from. So back to this. So basically what they've done is during these sessions, they train a decoder based on half the trials, and then they're using that decoder on a test, a withheld data set, and they're showing that just looking at this coding direction vector, so this vector that gives you a signature of is this distinguishing the neurons that are active during lick right or lick left, that it's very accurate at detecting in other trials, and indeed many days later even, whether the animal was licking left or licking right. You can see represented up on the right, top right, just that this works across long time periods after the animals had varying degrees of training. And on the bottom right, you can sort of get a sense of the weighting on each of the individual neurons in this, where you can see that most neurons are in the center, so they're sort of low level of weight, and then some neurons towards the top right have more weighting put on them. So it's those neurons that are giving a better discrimination signature of whether the animal's licking left or licking right. Okay, so this is all still, we haven't switched the contingencies yet. Ken and then John.
Before the switching of contingencies, this is probably an important question. naively, what I'm looking at here says that there is no representational drift. Although I don't know what I don't follow that literature that much either, although I know it is very in fashion to say that there is a representational drift. So saying that this is able to decode, even though you've trained it on an early and tested it on a late versus trained it on a late versus tested on an early, naively, that seems like, hey, there's no drift. But there could still be drift. And I'm kind of just wondering, are they making the case that this is very outside of what other literature has seen that they call drift, and that this is really not drift?
So we're preempting the discussion a little bit. They are indeed saying that this is like at most very limited drift. And they will go on to talk about how they wonder if the drift happens in other areas of the cortex, but is much more restricted in motor cortex compared to where they've seen elsewhere. That's kind of why, if I really wanted to prep for this session more, I would have dived into that literature to be like, where is drift happening and where isn't it happening and under which conditions? And yeah, let's come back to that question towards the end, because there'll be a few more things to say about it. John, you had a question?
No, I didn't. I accidentally hit it. I'm sorry.
Okay. No, it's all good. All right. Okay. So now the question is, what happens when we start switching the task contingencies? So the animal, it's gotten really good at licking left if the pole's at the front, licking right when the pole's at the back. And then suddenly we're mean experimenters and we switch the contingencies on the animal and they get confused and they have to learn how to do the task again. What happens in their motor cortex when we do that? Well, to show you here, what happens if you try and use a decoder, if you try and look at the activity with the neurons being sorted by how they were firing for the sample, the delay, and the response in context 1, but then you use that to look at the neural activity in context 2. So you're sorting by context 1, but you're looking at the neuron activity in context 2. What you see is that the same pattern is no longer encoded. So it seems to be basically randomly scrambled. If you're sorting across different contingencies, is what you see in these heat maps. And if you look at the bottom here, this is a representation of neurons that were selective during licking left or licking right during context 1. That you can see on the left here. And if you look at those same neurons on context 2, and they're now scrambled for whether they are firing based on licking left or licking right. So this is just to say that the encoding during the delay period for whether the animal should be licking left or licking right by these neurons is not being kept across these different contexts. Now note, again, this is for the delay period, not necessarily for the sampling of the responding. This is just to show that in a decoding fashion. So you can see that if you've trained a decoder on context 1 and then you're testing it on other data from context 1, you get these very high performances. And the same for if you've trained it on context 2 data and you're testing on context 2, very high performance. And you get, in contrast, almost chance levels when you're doing it from training on 1 and looking at 2, or training on 2 and looking at 1. Although with quite a lot of noise as well, which is interesting as well. So some of the individual decoding accuracy seems to work well, but on average, it doesn't work very well when being analyzed across contexts. This is just to sort of give you a sense of what it looks like if you're using the decoder for neural activity at each individual time point during a session, where you can see if you've got this coding direction delay vector that you're using as your analysis. So you're basically saying how much does neural activity at any particular time point when projected onto the activity that we know encodes licking left or licking right, how much does it correspond to that? You can see that at baseline, before any individual trial, it's not projecting very much at all. During the delay period, it either projects onto it a lot if it's licking right, or negatively if it's licking left. and then it goes away again as the trial finishes. This is if you're doing it from context 1 on training data onto test data. And this on the right is just to show you that it really doesn't work if you're doing it across contexts at these various time points. John?
That seems so interesting that in the second two, the individual neurons seem to spread randomly across the, like a very large field. Do you think that that's because that's just some Gaussian thing, or do you think there's something possibly going on where the neurons are trying different decodings and they're all kind of spreading out some way? Like, how would that? I don't even know how that would work, but I just find that really interesting that they're not grouped; they're just kind of spread out.
Yeah, I don't have a very good sense for why different. I even trying to remember what the exact unit here is in this decoding accuracy. I can't remember whether it's different because I think they often used as their unit of decoding the different fields. So like in each mouse, they had different fields, like different windows over the motor cortex. And within those different fields, they had like a set of individual neurons. So there's maybe like 200 or 300 neurons for each field. And there's maybe like 6 or 8 different fields per mouse that they're looking at. And it could be suggesting that maybe a set of neurons in each one of those fields is more encoding even during the delay, whether it's going to be licking left or licking right based on something else. Or I'm honestly not sure. I'm just speculating.
I found this very interesting as well. So there's 10 mice here, and there's 58 viewing areas, I guess. Each one is a dot. It would be very interesting to know if some mice were, treating it the same and some mice were treating it differently, or if this is an area-specific thing. They didn't say anything like that?
No, they do go into in quite a bit in the extended figures how the mice seem to be responding in different ways and how the field seem to be responding in different ways in a way that suggests quite a few different strategies are going on both per like individual mouse aggregated and in possibly these different fields. So I didn't go into that in as much depth as potentially we could. Depending on how we're going for time and whether we're still interested, I might pull those figures up at the end. But it's a good question. So yeah, this was the effect of new learning, basically showing that the neurons remap basically when they go from context 1 to context 2 in a way that the authors point out is reminiscent of place cells in the hippocampus remapping across different contexts, where in a different context, those neurons, they start doing different things. But now what's particularly interesting is what happens if you then go back to context 1. So you've trained an animal, you've gotten it to lick left for the pole that's in front. Then you've been mean, you've switched the contingency on the animal, and then later on you're mean again and you switch it back to the original context. Are you going to see just again a remapping of those neurons in the ALM so that their activity, re-encodes the contingencies of context 1? But is it going to be in the same way as when they originally learned the task, or is it again going to be completely random and you can't use a decoder that was based on the original context 1 activity to decode again in this context 1 prime that's happened after it's done a couple of reversals? Or will it work? Is the question that's being asked here. And again, this is like 2 months later now as well than when they originally learned this context 1. So initially what we're showing here is, again, just the neurons sorted by their activity during context 1, during the time for each individual trial. So you can see neurons that are firing during the sample, the delay, and the response period. And we're interested in these neurons that are firing during the delay period. And what you can see is when you go from context 1 to context 2, it's kind of a bit scrambled. But when you go from context 1 to context 1 prime, you see that it looks pretty similar to what the neurons are doing during context 1 and context 1 prime. And again, if you look below at the neurons that are firing selectively to indicate lick left or lick right during these delay periods, context 1, this is the reference data. Context 2 looks scrambled. Context 1 prime, not exactly the same as during context 1, but still pretty distinct signature of seeming to a large degree return to the behavior that happened during context 1. And then to show you this as a decoder, the top and the bottom images here are the same, by the way, just flipped around as to whether context 1 or context 2 was the reference. But what you can see is that a decoder that was trained on context 1 and tested on context 1 trials, which had very high accuracy, doesn't work for context 2, but does work again for context 1 prime. And that's the same whether you're using context 1 or whether you're using context 2 as your reference. So this is just to say, in summary, that it does seem to be the case that the activity of these neurons in context 1, again, after the animal has done a few different versions of the task, and after 2 months have elapsed, seems to be pretty similar to what it was 2 months ago when they were initially doing that task. And then this is the last set of data that I will show during these main slides, but we can pull up the extended figures if we're interested. What this one is asking is, is it something that's specific to this sensory tactile task that wouldn't come up if it was a different kind of learning? Or is it sort of robust across different sensory conditions? What they do here is they do another context 3, where the animal is, instead of having the pole moved around, they're having a sound played to indicate whether they should be licking left or licking right. And all this is showing is that the decoding for whether they're supposed to lick left or lick right. Hold on, now I've confused myself as to. No, yeah, they're showing that it still doesn't work if you're using a decoder based on licking left or licking right. If you're trying to go to a different context, whether there's, sorry, I confuse myself. I did have this yesterday, but I'm trying to remember why this data isn't particularly obvious, because what it's showing is that if you train a decoder based on context 1, it doesn't work for context 2 or context 3, whether context 2 was based on reversing the same task, and context 3 is based on now using sounds instead of tactile stimulation. And it's showing that, it doesn't work very well in each of those conditions. Oh, no, I remember why. But they're showing that decoders that are trained based on auditory, a task where the context is purely auditory, can still work with reasonable efficacy. So they're still getting 80% decoding when they're training these decoders based on context 3 and then testing them based on context 3. So it's not something that's specific to tactile sensation that's enabling them to do this task and to be able to do the decoding performance. But this is really more of a control sort of experiment than anything in particular. Anyway, I'll leave it there. Let's get into the conclusions and discussion. So I'll talk briefly and then I'll just open it up for everyone to discuss. So the conclusion here is these preparatory representations are stably encoded by the same neurons over multiple months. So they're robust to unlearning or reversals, but they seem to be context specific. The authors indicate that this reorganization of motor neural activity in different contexts resembles the place cell remapping in the hippocampus, where you see neurons that are indicating location in the hippocampus remap based when an animal's in various different environments, even though they're stable for the same environment for the same animal over at least days. And some of the bonus material that's in the paper that I've left out that might be of interest to some of you is they also do some computational work where they try and make a computational model of what's happening in the motor cortex, where they argue that it's better represented by a feed-forward network than a recurrent network. I didn't find this particularly compelling or interesting, but maybe you would. They do show that there is representational drift in task irrelevant activity, which I found interesting. So basically what they do there is they look at, they deliberately sort of like factor out the activity that seems to be selecting for licking left versus licking right. And then they look across the different contexts over time to see whether there's drift in other stuff that's not encoding licking left or licking right. And they do see that there's drift in like the other random activity that's happening over time. So it's not that the neural activity is like literally the exact same signature at different sessions across the months. It's just that the neural activity that seems to be encoding licking left or licking right is staying the same. And the other thing they also show is that there is a little bit of a change in the exact signatures of these coding directions based on whether they've learnt a task previously or not. So it's not that the activity remains exactly the same. It does get influenced to a small degree based on how many reversals they've done, for example. A couple of questions I have based on this. One is, we've sort of already discussed this, is like, what is it that's leading to us to see representational drift for some representations in some cortical regions under some task contingencies or sensory conditions or particular experiments, but not in others? And then the question that I think we'll get more into in a moment is like, what is actually encoding these preparatory representations in the motor cortex across months and across context shifts? One technical question I had that I'm just curious if anyone knows the answer to is like why you would use channel rhodopsin to activate GABAergic neurons rather than just photoinhibiting excitatory neurons directly. I don't know whether this is a practical reason or a sound technical reason. It's just if anyone's got more experience with these optogenetic experiments, I'm curious. I'm seeing a head shake from Ken, and probably nobody else knows. Anyway, just one question that I had. And then lastly, just this is my last slide, the relevance to memory decoding. So the main takeaway I have from this is, if I was trying to think of an experiment to do memory decoding based on looking at preserved static structure, I feel like we've seen quite a few papers now that indicate the motor cortex might be a good place to go hunting, because it seems to have these quite stable representations of information across long periods of time, where in this study we're showing that it's robust even if you train an animal on like multiple quite related tasks. It's like basically the same task with reverse contingencies. And we've seen these previous studies that show, for example, that after learning a particular task, you have these spines that are retained over months in the motor cortex, although that's in M1 as opposed to ALM. And we also did another Journal Club paper earlier this year showing these distinct synapse clustering in the motor cortex when the animals are learning these tasks. And unlike in the hippocampus, these things aren't getting thrown out all the time. Although I know it's contentious to what degree the hippocampus is holding on to information over long periods of time, as we've done a few journal clubs on now. And that is it. So I will open this up to general discussion as to what people think. So does anyone have any questions or thoughts on this sort of work? Otherwise, I have some more questions to throw out there, but I will start by asking if anyone has their own question. We'll go with Kunal. Sorry if I'm mispronouncing your name, and then Ken.
No, yeah, that was correct. Yeah, I mean, this was actually on the computational model, which is something sounds like wasn't of too much interest to you, but so I guess it's just general and open one. I couldn't quite figure out what was the input that they were providing to the models. Is it the activations of all the neurons, or was it just some basic parameters in terms of the pole position, etc., that they were providing? Did you get a sense of how detailed they were going with the computation model?
I will be honest, I read over that section, and then when I didn't find it super helpful, I sort of forgot a lot of the technical details. I think they were providing it though with the neuron firings at like at each time point for all of the neurons rather than just like the pole positions. But no, I honestly don't remember. I could be talking shit. Sorry.
No worries.
Ken, you're muted again.
This might be a question related to modeling as well. I didn't read that part of the paper. But it struck me as kind of weird that if I thought about how I would do such a task, folk psychologically, I might during the sense period, I might say, "Oh, I saw the bar there. That means I got to lick left." And during the delay period, I'd be saying to myself, "Lick left, lick left, lick left." I wouldn't have the whole context of the experiment in. And so it's weird that, to me, that it didn't just track with the motor output that was going to be going on or with the input, that it was literally a signature of the task as it will, even during the delay period. Did they, or do you have any kind of sense of what we're supposed to think of that preparatory activity actually doing?
Yeah, that's a good question. So why isn't so they show the response activity and they show that's always the same. Basically, you see the same activity driving licking left and licking right when the animal is actually like licking left and licking right. And during this delay, why isn't that the same across different contexts? So why aren't they constantly, even during this delay, they're like lick left, lick left, lick left, lick left. So presumably it's like, they're like, which one am I supposed to do? Am I supposed to be licking left or licking right? Based on this is maybe what that activity, maybe it's like, they don't know yet whether they're supposed, because during the sensation, it's like, that's when they're like, okay, the pole is in the front or it's in the back. And then during the delay is they're trying to decide whether it's lick left or lick right. And that's what the neural activity is encoding at that particular point in time.
So presumably if they had lengthened that delay, then and this gets back to John's question, I think. If they had lengthened that delay to like 5 seconds, then maybe the last second in that delay wouldn't have reflected a thinking about the task. It would have reflected I'm really decided to just lick left. Anyway, yeah. I think I've got the answer. Thanks.
No, that's a good question and relevant to something I've been wondering about, which is like the interaction between working memory and long-term memory in these experiments. So obviously, like there is some long-term memory and that they have to remember based on their sensation, am I supposed to lick left or am I supposed to lick right? And that's something that they've had encoded over multiple days and multiple trials. So it's not purely working memory for everything. But obviously this task is very working memory dependent in that it's like, they've just had the sensation on their whiskers and they're trying to decide whether they're licking left or licking right. And they've also probably done like a lot of trials in a row, given any particular trial. So there's some short-term memory contribution there as well. And so I get a bit confused when I'm trying to imagine how to disentangle the like long-term memory components from the working memory components during any of these individual trials and their activity. But I see Kunal has a question.
Yeah, no, actually, I was just going to comment on the discussion you guys were having. My sense was that this wasn't as much about the long-term memory. And when I read the paper, I didn't think the 1.3 seconds was had sort of any meaning other than my thinking was that when they are recording, there's just constant firing going on, which is why and the mouse would just be licking constantly, which is why they trained it that only after this delay cue is what you're going to act and you're going to be rewarded based on that. And that would make their recording a bit easier, right? Because just around that period is when they essentially record data. That was the sense I got, that it was more in terms of just the data they have, and just an easy way to just track, okay, you've been trained, it's after this that you're going to be rewarded or not rewarded, so do it based on that.
Yeah, I'm not sure whether they penalized the mice for licking early or not. That was, it didn't mention it specifically in this behavior, but I think they did say that, like, they referenced an older paper, which I didn't go check, about how they did it. So I'm not sure to what degree the mice were licking or not before the response was elicited.
Right. I guess they just won't get a reward, right?
Yeah.
They just won't get the water otherwise.
No. So. John?
Yeah, thank you very much. What a wonderful test bed this seems to be. Do you get a sense, or did you get a sense that there's new ensembles being created in motor cortex for the new tasks, and that the original tasks are still there as a separate set of engrams? And a related question, was there any difference in the speed of reacquisition of the 1' over 1? So those would be my two questions.
Yeah, your second question first. There is an increase in speed. They do start to learn the reversals faster. And there's also variability within the individual mice as to how quickly they start to pick up on the reversals faster. And there is also some relationship between the variability and how quickly the mice pick up those reversals and the representations that you see that they have in the cortex, where there's sort of a bit of a signature of the mice that pick up on the reversals faster.
Great. So a follow-up question then. Do you think that they're basically using something, I'm probably not using the right phrase here, but kind of silent synapses within the existing network to encode the additional contexts? And would there possibly be a maximum of which they could do that if it's a kind of a subtractive process, let's say, that's still connected to the existing one? Do you think you might be able to test kind of the limit of that with something like this?
So given they're going backwards and forwards rather than trying to test multiple at once with different like interleaved contexts, I don't think it gives us much of a sense of like how many you could pack in. Maybe you'd see, I mean, if you're seeing that like there's accelerated learning of the reversals, maybe what you'd see over time if you tried to train them on too many tasks, different distinct contexts, is like it would just plateau how quickly they could learn the new one. But I don't have a good answer to your question there. In terms of your first question around, you know, what's going on with the ensembles? Are they distinct ensembles? I think the fact that they're showing the same neurons that are active for the same task across months, and those neurons get remapped if they're doing a different context, and then they come back on in a reliable way if they go back to the same context, does seem to indicate that, like, yeah, there's a pretty stable ensemble that represents the task contingency across time, and that they're different.
If I understand that right, what they're basically showing is roughly the same neurons, but the ensemble is something that's a sub activity within the neurons in the remapping. Is that correct, or are those new neurons in the new context in addition?
I think they're tracking the same neurons across the entire experiment. So it's like the ones they pick up on day one, session one, they've identified in each of these fields, and then they're just like looking at their activity throughout the rest of the experiment, even if that's 2 months or 3 months long. I think they threw out neurons that they couldn't reliably track over time.
So then is your intuition then that basically what they're doing is they're kind of mapping different ensemble capabilities within the same neurons using something in the cyto architecture of those neurons? So like they're tapping different spines and different kinds of integrations, maybe different synapses.
I think the fluorescence they're picking up on, and again, other people can correct me if they know more, would mainly be from the soma of these neurons or from their cell bodies, I think is what they're tracking. So it's more that they're just looking at how the activity changes for the same neuron across different task conditions, rather than that they're looking at subsets of parts of the neurons.
That's very helpful. Thank you. Yeah,
different conditions. Cool. I see we're a little bit over time, so people are welcome to stay and discuss if they want. But otherwise, thanks very much for coming. We'll have another talk in 2 weeks or so. We're starting to get close to the satellite event. If anyone else wants to present a paper at any point, then they're very welcome to do so. If anyone wants to start suggesting some nominations for papers that should win one of the annual awards this year, that would also be super helpful. You can either email it on to me or to the group mailing list, or you can submit it on the website. But thank you very much for coming, and I'll stick around for a minute in case anyone wants to say anything else or do anything else.
Thank you.